Please welcome Dr. Peter Marrin Kovich.
Hi, everybody. I have the same problem that Al Lane has as far as the height of the microphone. Can you hear me okay? Yeah, sorry about being outside. I just got involved with talking to some of our EB patients out there. You guys are really fun to talk to. Anyway, I'm very happy to present some of the work that we're doing at Stanford in collaboration with Dr. Al Lane. And he and I are doing a number of different therapies along with a very talented team of individuals who I'll introduce to you later. So today I'll be talking to you about two different approaches that we're using to treat epidermolysis bullosa, and currently we're focusing on recessive dystrophic epidermolysis bullosa. I don't think I need to give you a detailed description of this disease. I think you know it, you're very familiar with it already. You know, it's a devastating disorder, and it's in desperate need of a specific molecular therapy. It's extremely frustrating for the patients, for the families. It's even frustrating for the physicians too, because we see the disease process progress, and we're so limited currently as to what we can do. So the defect in recessive dystrophic epidermolysis bullosa is collagen 7. It's what we call a basement membrane protein. And all the different basement membrane proteins, they connect together like little links on a chain. And type 7 collagen is at the bottom of the links of the chain around here. So when this is absent or defective, then this whole little connection apparatus, we call it the attachment complex, falls apart, and then you get blistering. And you get a kind of, because this is the deepest part of the basement membrane, when you get the blistering, you get a lot of scarring. So it's an especially deleterious process. So how do you correct epidermolysis bullosa? Well, first you have to understand how it takes place. And that's taken a number of years. First we had to discover the basement membrane's own proteins, then we had to discover the genes, then we had to characterize the mutations, and finally we're now at the stage where we're able to start to correct the disease. So first you have a mutation in the DNA that gives you an abnormal transcript, we call it mRNA, and that codes for a protein, but the protein is defective because it comes from a defective transcript. And then that protein is being used by a cell who can't use it properly because it's defective. And then the cell is in a tissue, and then when you have the disease cells in the tissue, that's when the tissue falls apart. So there's a couple of different ways that you can correct this problem. First, you can take the gene and go right at the DNA level and then correct this portion. And that's in the first part of the talk. I'll talk about how we take the gene and correct it. And then another way, a little further down the road here, you can take the defective protein and put in good protein to sort of make up for the lack of the bad protein. And we'll talk a little bit about that at the end of the talk. So first off, let's talk about the gene-based transfer. And in this process, we have multiple steps. And I'll just walk you through it. So I think it's really important that you understand exactly what we're doing here. So first you get a skin biopsy from the patient, and from the skin biopsy, we can expand the cells in culture. And then next, we take the DNA, the correct gene coding for type 7 collagen, and we deliver it to the skin cells so that all the cells or majority of cells are producing the correct gene. Then we have to grow the genes into sheets, and then following this, we take the sheets and graft it back to the patient's skin. So that's sort of an overview of what we're doing. So initially, when we culture the skin cells, we basically take them out of the skin, we grow the keratinocytes, which is the outer layer of the skin, and these are the cells that produce a large amount of the type 7 collagen that we use to hold the skin together. And then we use this gene delivery device. It's called a retrovirus. So it's basically think of it as like a DNA vehicle where you can put the type 7 collagen gene into this, and then you can use this virus to infect cells. And it very efficiently, along with infecting the cells, it carries the gene along with it, and it incorporates stably into the chromosome. So what that means is that there's a lot of other gene transfer techniques where the DNA is not incorporated into the chromosome. So that means that every time the cell divides, the DNA is lost. Like, for example, gene gun will shoot the DNA into the skin, but it won't incorporate it into the chromosome. So after a day or so, the gene expression goes away. In this instance, when it goes in the chromosome, every time the cell divides, there's a new copy of that therapeutic gene that goes to the daughter cells. So that's, theoretically, this should give us extremely long-lasting gene expression. And then what we do is we take the virus and we expand the virus to make large amounts of it to support large grafts. And when you're working with viruses with the FDA, you have to be extremely careful, and you have to follow a lot of guidelines and do things in a very correct fashion. So we have to develop a very efficient protocol for every step in the production of this virus to make sure that it's extremely safe. And then the next step is to, once you've got the virus and put it into your keratinocytes, next step is to make the sheets. And the first step is you have to have a really clean facility to be able to ensure sterility and safety so that you can ensure that the grafts that go onto the patient are as safe as possible. So we've actually designed our own facility at Stanford. This is a schematic of it, and it shows that we have these filtration units which filter out all the harmful bacteria. We have a number of other chambers where you go through to ensure sterility going from one area of the unit to the other. And then in addition to making the actual production facility, we also have to put together a very detailed protocol. I'm not going to walk you through all of this, but I just want to show you this just to kind of demonstrate how detailed you actually have to be going through every step of this process in order to finally get what we call release of the graft so that it can go on to the patient. So it's an extremely prolonged process here, so that when we do our grafting, it takes pretty much all day to go through all of these steps before we even get to the chance to be able to put the graft onto the patient. We're looking for different kinds of infection. We're looking for different kinds of viability, or how well the cells are alive. We're looking at harmful pieces of DNA which might cause complications in the grafts. So after all that's done, then we can actually take these grafts, and this is what they look like. These are the skin cells, and they're all grown together in a little sheet. It's about 3 or 4 cell layers thick. And what we do is we take a piece of a nonstick dressing, we put it on top of the sheets, and then we pull the whole thing off. The nonstick dressing kind of static clings to the keratinocyte, the skin cell sheet, and it comes off, and then we staple the sheet of the keratinocytes to the nonstick dressing, and then we carry that into the operating room. So once you've made the gene and packaged it and developed the protocol, developed the room to make it, got all that done, can make the sheets, next step is we have to design the protocol. And this is where Dr. Lane's expertise has come in. He's very good at this. And we've produced a number of goals here. One is to be able to show that we have efficient expression of type 7 collagen in the short term, which would be 12 weeks, and then the long term at 6 months. We want to be able to show that the type 7 collagen is positive by microscopy analysis. We want to be able to show at the super high magnification electron microscopy that the type 7 collagen is functional, and I'll show you some pictures about that. And then we also want to make sure that this replication competent retrovirus, this is a harmful complication of retroviral therapy, which can produce complications such as cancer. We have to be extremely thorough in ensuring that none of that is present. So with those goals in mind, we've been selecting patients for the trials. First, we screen them to see if they're the best candidates, and we look through inclusion criteria. What are the good features that we're looking for to have the best chance of success? And then we also look for exclusion factors, which are factors which might predict a poor outcome. And by the way, we are screening patients currently, and we're hoping to be able to expand these trials in the future. Currently, we're working with adult patients, and we hope by the end of the year, early next year, to be able to progress to working with children. The FDA feels that it's the most ethical way to proceed is to work first with adult patients because they can give full consent. But then once we're able to show without a doubt that this is a safe and well-tolerated procedure, then the FDA feels it will be applicable to be able to work with children. So we're moving towards that goal. So we're screening both adults and children, anticipating we'll be moving in that direction next year. So this is the results, very exciting results from our first patient. And what we were able to show was initially on the screening, we found that this patient has almost undetectable type 7 collagen. If you look by the microscopy, you see it's absent. If you look by even the high magnification microscopy, this electron microscopy, it's also absent. But if you look very carefully at the proteins that the skin cells are producing, you see just a little bit of protein here. And this is good. At least to begin with, we don't want to have a totally negative protein. And that's because the patient's immune system, if they have absolutely no protein at all in their body, the patient's immune system, when we give them type 7 collagen, might recognize the collagen as a foreign protein and develop an immune response. And we want to avoid that, at least in our first few patients. So all of these criteria fit very nicely as a very good inclusion criteria to proceed, and that's what we did with this patient. So we selected a number of sites, areas of chronic wounds, as well as we actually induced a wound on one of the areas of skin. And then we made all of these grafts, and we analyzed each of the grafted sites following grafting. This is before at 12 weeks and 25 weeks. And you see exactly where we grafted, there's persistence of the graft and absence of erosions. Particularly striking was the shoulder. The shoulder was an area that the patient had a very long-term chronic erosion on. I think he slept on that side. And he doesn't remember a time when his shoulder was completely healed. But as you can see here, there's maybe on the edges of where the graft, the border of the graft with the erosion, there's a little bit here and a little bit here. In the actual square area that we grafted, it still looks beautiful. The patient's very happy with this result. So looking at the microscopy, you can see that the type 7 collagen is restored in these specimens. And we also looked at some of the other proteins that are important for the skin to develop properly, the differentiation proteins, which give a nice barrier to protect the body. And all of these proteins are being expressed correctly. So it shows that not only is the type 7 collagen gene being expressed in the skin just as we had hoped it would be in a long-term fashion, but also the skin is developing properly and behaving just like normal skin. And this is that electron microscopy. And what we're able to do is we're able to take tiny beads that have an antibody attached to them. They're made out of gold. And wherever the gold is present, that means the type 7 collagen and an antibody is present together. So you can see the uncorrected skin has none of these little gold particles. But the corrected skin does. And you can see the gold particles are wrapping around these little lines are dermal collagen. And that's what the anchoring fibrils do, is they wrap around the dermal collagen. You can actually see these little beads wrapping around the dermal collagen here. That's exactly what type 7 collagen does. It wraps around the collagen. So just from the appearance, it suggests a functionality to the collagen. So the conclusions from this therapy so far with this initial patient is that we've been able to develop this good manufacturing practice or clinical grade retroviral vector. We've initiated our phase 1 trial for correction of RDEB. We've grafted our first patients successfully. We've been able to show robust and persistent type 7 collagen expression. And so far, we're monitoring closely for processes like autoimmunity and cancer, but so far, the patient's doing very well. And this just to reiterate, the problems we are looking for are immune reactions to type 7 collagen, development of cancer, and of course, more short-term problems like infection and sloughing of the grafts. So this is where we stand right now. We'll be continuing to work with a number of other patients with this identical procedure over the coming year. But we're already looking towards the future as to how we can further refine and improve this process. And one of the things that is at the forefront of biomedicine currently is this idea of the induced pluripotent stem cell, or iPS cell. This is a cell that can be, you take a normal cell and you let it go back in reversion back to the primordial embryonic state. And then from that state, you can get unlimited potential for cell division. One of the problems with this current method that I just showed you is that we have to grow the cells up pretty darn quick and get them on the patient before the cells run out of their proliferative potential. So it would be really helpful to be able to have cells of unlimited proliferative potential. We could, from a single biopsy, we could graft an entire body and have cells to spare. So that's what this protocol details, is how to make the induced pluripotent stem cell. So you biopsy the cells and culture them, you reprogram them back to the embryonic state, and at that point, you put the correct gene back in, and then you can bank them and hold and keep them in place just like a reagent, just like a medicine on the shelf that you can just pull out as needed. And then once they're expanded, then you have the chance to select them to be able to weed out the cells that you don't want, and that can be a very helpful process. I'll give you an example in a minute. And then finally, you can just graft them as a sheet back onto the patient, just like I showed you we did with our first patient. So right now, this is only at the animal testing state, but one of the things that we have been able to do is to show that we can use this technique to weed out cells that may have some markers for future squamous cell carcinoma. We know, all of us appreciate here that this is a serious problem for recessive dystrophic EB, and if there's a way to be able to remove cells that may have an increased potential for having a cancer, that could be a very good thing. So what we've been able to do, and this is actually in the lab of Dr. Tony Oro, one of the colleagues of Dr. Lane and I, and he's been able to show that but in this selection process, he can detect which genes may be showing an increased risk for cancer, and then he just throws those cells out. And he selects the cells that don't have any of these gene risks. And he's been able to show, grafting it to mice, that he's been able to remove some of the potential risk of cancer during the grafting process. And this just shows, following grafting to mice, how you can get a nice, robust type 7 collagen corrected expression here at the basement membrane of the grafts. These are on, again, these are on mouse. This is not a clinical trial yet. As well as a good nice structured epidermis, or outer layer of the skin. So this is this potentially could be a future refinement of the current process that we're doing right now. So, this is just a stepping stone along the way to be able to develop the best molecular delivery to correct recessive dystrophic EB and ultimately all the different forms of EB. But there's things that it can do and there's things that it can't do. For example, it's hard, technically, you can appreciate that it would be technically difficult to be able to place grafts into the esophagus or other areas that are constantly being used because you'll get the grafts sloughing off. And then there's other therapies which potentially could be complementary to this. And one of them we feel is protein therapy, where you actually take the protein and deliver it into the skin. And I'll tell you a little bit about some of the studies that we're doing in this area to close the discussion. So basically, as I mentioned before, type 7 collagen is at the bottom of the basement membrane. It likes to loop around the collagen fibrils of the dermis. There's like one collagen likes to loop around another collagen. And if it doesn't work properly, then you get separation of the skin. This is the basement membrane here, and then here's the basement membrane falling apart in EB. This is what the type 7 collagen protein looks like. And it forms a little trimer, or 3 molecules together. We call it a triple helix. It's sort of like a coil of rope. And that's what collagens are. They're like little rope coils. And then what happens is you get one coil of rope getting together with another coil of rope, and they form a double coil. We call it an antiparallel dimer. And then once that forms, then that precipitates this reaction where a lot of these antiparallel dimers cluster together, and that's how anchoring fibrils are formed. So when you're making the collagen, you want in the culture before you put it in the skin, you want it in this state as much as possible. Because if in the test tube or the syringe before you inject it into the patient, if this happens, it's going to clump up and it's not going to get to the place that you want it to be. So you want to be able to keep the collagen 7, what we call soluble, as much as possible, prevent that anchoring fiber formation. Collagens also are very specialized molecules. They require other enzymes to sort of process them and make them as good as they can be. And one of these proteins is called Prolifera Hydroxylase. You can kind of think of it as reinforcement to the rope. It keeps the rope from unraveling. And in the skin, when the collagen unravels, it gets degraded. There's proteases that are always maneuvering around trying to chew the collagen up. And they look for any little opening. And when there's a little unraveling of the collagen, they go right in there and chew it up. So if you can avoid the unraveling, you can make the collagen last longer in the skin once you put it in. And that's been one of our goals. We feel that this Prolifera Hydroxylase, if we can increase the activity of this enzyme when we're making the protein, we can allow for a more durable protein. So instead of having it last only 2 months after we inject it, we hypothesize that it could double the half-life, maybe make it last 4 months. So as a first step in this protein therapy, we have to make cell lines that can produce large amounts of collagen 7. And we already have this technology in biotech industry when we're making antibodies. All of these biological antibodies that you see on commercials, Embrel, et cetera, they're made by these little cell lines, CHO cell lines, that pump out large amounts of human protein. So we've made a CHO cell line that pumps out large amounts of human protein, and we can show that it produces protein from many passages, 18 passages, which is more than enough to go through a full production. And then we have also put in that Prolifera Hydroxylase that makes the collagen more stable. And what we found is that the cells, these CHO cell proteins that have expression of the Prolifera Hydroxylase, we have this assay where we take these proteases and we look to see if, when we heat the collagen up, are the proteases chewing up the collagen. It kind of simulates what might be occurring in your skin when the skin becomes warm. And the idea is that as you heat the collagen up, it starts to unravel if it's not very stable. and then the proteases chew it up. So as you can see, at the 40-degree time point, at 38 and 40-degree time point, the Prolifera Hydroxylase collagen is actually very resistant to the enzymes. But without it, it does seem to be digesting. So this is just the first step towards establishing that the Prolifera Hydroxylase expression is actually helping to make the collagen more stable. But we think it's a move in the right direction. Next, we have to sort of engineer our cell line to fine-tune it to make as much collagen as possible. So there's new refinements in cell culture production that we've borrowed from industry, the so-called fed batch culture, where we grow the cells for 14-day cycles, and then we add things that the cells like to grow in: glucose, plant hydrolysis, which is basically just adding extra protein to the cells, and the special feeds that they like. And then we monitor things like the density of the cells over the course of this 14-day cycle, and we try to keep the density, get the density up and maintain it. And we look at viability of the cells. We also look at the glucose concentration, which is it correlates very well with the cell viability, as you would expect. So the final product is you get a nice pure collagen 7 molecule, which then you can inject in the skin and get a nice deposition of type 7 collagen into the skin here. So it's been well established, not just by our group, but also by other groups, including the UST group with David Woodley. It's shown that if you inject type 7 collagen in the skin, it will diffuse right into the basement membrane where it's needed to be able to help to correct the Epidermolysis bullosa defect. So what we're doing is we're taking this purified collagen 7, and instead of just injecting it with a regular needle, we're using microneedles. And these are little, very tiny needles that are about the thickness of the epidermis that go just down into the skin, just enough to dissolve, and then when they dissolve, they release the type 7 collagen just at the level of the basement membrane where it's needed. There's other protocols out which are like proposing to inject collagen 7 via like intravenous therapy, and they also show some promise as well, but we're appreciating that the collagen 7 is very expensive to produce, and we feel that this microneedle approach is going to give us the most bang for the buck as far as the amount of delivering the collagen in the most efficient manner. And we also envision that the advantage of this microneedle is that the collagen microneedle patches could be sterilized and then put into wound care-like dressing packages, and you won't need a specialized center to transfer an epidermal sheet or give an infusion. You can these patches should be able to be applied just by home givers at home in a fairly non-technical fashion. So this is the normal thickness of the skin. You see that it's about 50 to 300 microns. It depends whether we're looking at the palms or looking at the eyelids. The thickness of the epidermis varies. And so we design our microneedles to accommodate for this thickness. This is what they look like. You can vary the length of the needles. You can vary the amount of collagen you put in the needles. And then you can also vary the spacing of the needles. So in this way, by these three parameters, you can optimize the collagen 7 delivery in a very precise manner. So we've been doing work mainly in the laboratory on skin sections where we put the microneedle on one side and then we attach it to skin that we have immobilized on another plate. And what we see is after about 15 minutes, we get full dissolving of the needles. And then when we label the needles, and right now we're working with collagen 1 because it's cheap and easy to visualize. So if we label the collagen 1 with this red dye, we can see that it goes down below the epidermis here. And it is delivered, here's where the basement membrane would be. It's being delivered just in the right place. Here's just another photograph of it. So these microneedles hold promise as an easy-to-use, efficient, and hopefully relatively inexpensive method towards correcting recessive dystrophic EB. And we think it may be a nice complement to the gene therapy approaches that we've already been using in a preliminary number of patients. So I would just like to thank the rest of our group here, Al Lane and Zurab Sifashvili, who was really leading a lot of these efforts on the epidermal sheet production, Tony Oro, who helped out with the IPS cell work, and a whole host of other folks, without whose participation none of this would have happened. Anyway, thank you, everybody.